So welcome everybody, welcome on Endoscopy On Air. It's my great honor and privilege to introduce this talk altogether with Mr. Amyn Haji. Amin is, first of all, is a friend, so I'm so glad that I have Amin with us, but he's a super expert. He's a well-known expert in the field of colorectal disease, and he is a, would say, special profile, and I've always been a little bit envious about this special profile because he can go from both sides, endoscopical and surgical, so it's very interesting. So, Amin, very warm welcome to Endoscopy On Air. So glad you're coming and you accepted to be part of this talk. So we are connected from London and King's College. So please, can you introduce yourself?
Thanks, Ali. Great honor to be here, and thank you for inviting me. My name's Amyn Haji. I'm a colorectal surgeon. I'm a laparoscopic colorectal surgeon at King's College Hospital in London, and I'm also an interventional endoscopist. So I perform both upper and lower GI endoluminal surgery, and I've been here at King's since 2010. But I have a particular interest really in minimally invasive surgery and natural orifice surgery. And I think the link between laparoscopic surgery and interventional endoscopy now is growing a bit closer. Interventional endoscopists are being very much surgeons in their field. So this is a very exciting time for me in terms of development.
Okay, thank you. So from King's College, London, it's interesting because you say the laparoscopic surgery, minimally invasive surgery, transoral or trans-orifice surgery. So I'm curious, what is the difference? So I get confused. So you do any kind of surgery, basically. What kind of instruments?
Yeah, I think, I think now we're pushing the boundaries more and more. The way I like to think of it is organ preservation surgery. And that is really, can be from very different means. But the particular interest of mine is using endoluminal surgery, so either going in, transorally or transrectally to perform incisionless surgery. So particularly if we're doing endoscopic submucosal dissection, or if we're doing POEM or GPOEM or any metabolic endoscopy, this is all natural orifice and avoids the patient losing their organ, and also avoids the patient having any incisions through the abdominal or chest cavity. And that's a huge advantage for our patients in terms of recovery. So I think whenever we speak about it at King's, we always talk about organ preservation and how we can achieve that, and what is the best approach to try and achieve that.
Okay, that's a very good approach. So, I mean, this is quite unique because normally in the world of endoscopy, we have people with expertise only in the field of endoscopy, and the same is in the surgical space. So how did you get this hybrid expertise?
So interestingly, in my career, I was a budding laparoscopic surgeon. My interest was in laparoscopic colorectal surgery, but my research was interestingly in endoscopic ultrasound, particularly for assessing colorectal polyps and colorectal tumors using high frequency mini probe ultrasound. And as I was sort of doing ultrasound, we were examining a lot of polyps, and that got me interested in magnification endoscopy to assess these polyps in much more detail. And therefore, I decided to do a fellowship with Professor Kudo and Professor Inoue at Showa University in 2010, where I spent my fellowship year doing and learning about ESD and POEM. And at that time, actually, POEM was very new because Haru had just introduced it in 2008. So I had the opportunity not only to learn about magnification endoscopy, but also to learn about the techniques of ESD and POEM. And then coming back to London and to King's, it was really my responsibility to introduce that in 2011 to King's College Hospital. And then since then, we've really developed that practice and developed that unit, particularly now focusing on primarily on ESD of colorectal polyps, and also on treatment of achalasia with POEM and GPOEM, and expanding in the field of metabolic endoscopy. So it's actually interestingly, my sort of career path has moved from laparoscopic surgery and then doing research and then now moving very much into interventional endoscopy. But still I practice both, and that's been very interesting because sometimes I feel that you can offer the patient a sort of a view of what the advantages and disadvantages are of laparoscopic surgery versus interventional endoscopy, and then help them to guide them in their decision. In the UK, we're very much on patient-centered care and involving patients in their decision. And so this sort of fits in very well with that consent process and in discussing with patients, what are the advantages of laparoscopic surgery in your particular case, or what are the advantages of interventional endoscopy? So really excited by the development of this field.
That's good. If I understand well, you went to Japan to improve your skill in diagnostic modalities. You say the optical diagnosis, recognition of peak pattern, no, and so on. And you understood the value of endoscopic resection, no? Is that correct?
Yeah, absolutely. I think my main interest initially was on magnification endoscopy in the esophagus and in the stomach, and then more and more interested in colorectal polyp and magnification endoscopy. So it was really on the diagnosis side where I had my initial interest, and that led to really having the decision making for which patients would benefit from ESD, for example, and which patients could potentially benefit from EMR.
So coming back from Japan, how did you convince your gastroenterologist colleagues to to get accepted in the
Yeah.
space of endoscopy? So it was any problem, any fights, or just be honest.
It's
It's
very
a lesson to be learned.
yeah. I think it's really difficult because I think in the UK we're fairly unique because we surgeons actually do endoscopy, diagnostic endoscopy. Many parts of Europe, many places in Europe, surgeons do not do diagnostic endoscopy. But even then, actually, surgical endoscopy training is not as robust in the United Kingdom as it is in gastroenterology. So actually, it did take a bit of time to be accepted in the field of diagnostics. But fortunately, we have very good collaboration at King's with gastroenterology and colorectal surgery. So soon we actually got our gastroenterologists interested in magnification endoscopy and grew that field as a collaboration, and then really created a subspecialty of interventional endoscopy related to that. And now we have both gastroenterologists and surgeons that are part of that team, and we make decisions together and support each other in this. So I think that collaboration and close working is extremely important.
So do you think that for yourself, being a surgeon, the value that you had in the beginning was the fact that you can protect the gastroenterologist in case of complications of patients not be treatable by typical endoscopic resection. So having straight away ready to go another surgical alternative without going too much in discussion with the surgeons, or this was not a value?
Yeah, no, I think that was of huge value because it sort of it supports our gastroenterology colleagues in the rare occurrence that there are complications such as perforation or bleeding, and that patients may need to have laparoscopic surgery to support that, but that's quite rare. I think where the most value is really to support and give colleagues confidence and support their decision making that we would support our gastroenterology colleagues if there were to be a complication. So I think that had some value for sure. I think the biggest value was also to combine with our pathology colleagues and work together, particularly when we were doing magnification endoscopy, to look at the collaboration between what we saw with magnification endoscope and then what we saw histologically, and support each other in our own learning. Because initially with magnification endoscopy
Yeah.
in the UK, we had very little experience compared to that in Japan, and therefore we had to sort of learn our diagnostic skills and look at how accurate we were in comparison with pathology. So I think having that collaboration was fantastic. But certainly, I think supporting the gastroenterologist for complications was one way of being welcomed into the field of endoscopy.
Anyway, I would say this is a great story. I love it. So, I mean, if I can, I would like to move a little bit to the clinical space. And there is a lot of discussion about resection of rectal lesions, because of course there are multiple options, and mostly everything is based on the fact that you can properly stage the lesion, deciding whether or not there is high risk of invasive carcinoma or not. So what is your view? How you stage rectal lesions that are not obviously malignant?
I think this is, such an important topic, and it needs to be done well because the consequences to the patient of doing this badly is vast. For example, if we were to do a piecemeal resection, for example, of a polyp in the rectum, and there was incidental cancer, then the consequences to the patient would mean that they're likely to require quite major intervention in terms of surgical intervention to rectify the problem. So it's really important to get an accurate diagnosis, and we spend a lot of time with this. What we tend to do in our practice is use multimodal evaluation. So all our patients will have magnification endoscopy on assessment, and we would look at spectral imaging, look at their vascular pattern, and look at pit pattern in detail.
Is
Uh,
that easy when you have a very big bulky lesion? So is that possible? That's my concern all the time, that I'm missing a part that's not properly evaluated.
yeah, I think it's important to go with that in conjunction with the Paris classification and the morphology because it takes time to evaluate the surface characteristics of the whole
You
polyp.
think having a cap on the tip of the scope may help in focusing and assessing better?
Yeah, sometimes what we tend to use is we use a black cap, a
Yeah,
very
the black cap,
short
yeah,
cap,
very
which
soft.
is very soft, and then you can keep the focal length when you're doing 80 to 130 times zoom,
Hmm.
and evaluate in detail. When it comes to the nodular component, the magnification endoscopy is not as accurate on the nodular component as compared to the flat homogeneous part. So, although we look for surface characteristics, we can identify the high risk features. We need to do additional imaging in order to evaluate that. So, in our practice, we would use three modes. We would use magnification endoscopy, we would use high frequency mini probe ultrasound or 3D ultrasound to evaluate the polyp and look at the depth of the polyp invasion. And we would also use MRI as routine. And the reason we use all three, even if we feel that it is a benign polyp, the reason that we use all three, for example, people often say to me, why do you use MRI when you think it's a benign polyp in the rectum? And my answer to that is the first of all, we need to identify if there are any adverse features on the MRI. Are there any nodes? Is there any evidence of EMBI? Because if there is incidental cancer after resection, then doing an MRI after the procedure will always obscure your MRI images. You'll tend to get stranding and you may get some nodes and you may overstage the disease.
Can I make an evil advocate comment? So my perception is that most of these lesions, the decision is based on microscopic features, and still the reliability of the technology that we're using, whatever it is. So I mean, non-endoscopic, so ultrasound or MRI is not that precise that allowing you to take a decision based on true facts. And the other point is when you have a P pattern or vascular pattern that goes in the direction of the malignancy, that's a much more robust sign. So still endoscopy is predominant. Do you agree on that?
Yeah, absolutely. I think you can make the decision based on your endoscopic image the majority of time. What we tend to do, particularly, I mean, we were talking about rectal lesions, but for example, say if you have, five eye pit pattern in the colon, and we think on, and if it's JNET type 2B, and there may be an LSTG with a small nodule in the colon. Now, in our mind, we think the endoscopic diagnosis is likely to be high grade dysplasia, potentially, but this could be also early submucosal invasion. Now, with 5i pit, we're not 100% accurate because about 15% of patients that will have 5i pit will be histologically 5N. And therefore, what we tend to do in that situation is we may use, if we're uncertain in certain areas, mini probe ultrasound to see if the muscularis propria is favorable or not. Because if it's not favorable, then
Okay,
actually you may
good
want to make
point.
a decision, may make a decision regarding laparoscopic surgery. But if it is favorable, you could do a trial dissection with ESD.
So, having saying that, you think that all rectal lesions larger than 2 centimeters should undergo this kind of flowchart, diagnostic flowchart, or you have another approach? So, if it's granular type, non-macroscopic findings suggestive of malignancy, you just do endoscopic evaluation and that's it. How you do?
Yeah, so I
This
think
is must be a take-home message for everybody.
yeah, I mean, interestingly, Ali, it's a very good point that when we first started, we would rely very much on endoscopy. And if you had LSTG homogeneous type, and we thought that the pit pattern and the vascular pattern was favorable,
Regardless of the size?
initially, if it was a greater than 2 centimeter lesion, and if we felt it was LSTG homogeneous type, and we thought the pit was favorable, what we would do is we would just rely on the endoscopic diagnosis because the risk of malignancy in that patient is 0.8%. And therefore, we'd rely on the endoscopic diagnosis, but we would always do en bloc resection with ESD. We would never do piecemeal resection. We never do piecemeal resection in the rectum.
Wow, this is a big take-home message. So let me understand. Whatever is your prediction of histology, you go and block.
We go en bloc in the rectum. And the reason for that is in that 1% of patients that you may have incidental cancer in that LSTG homogeneous type, then the question is the decision making for that patient becomes very complicated because a piecemeal resection, you don't know whether the margins are clear, and you don't know whether there is residual disease.
Okay, but I have a trade-off. I need a trade-off. So this must come in a reasonable time. So it means cannot be with a 3 hours, 4 hours, 6 hours procedure first. And the other one is that with a very, very low risk of complication. Is that correct? Otherwise, it's difficult to say to everybody, okay, just do and block.
Yeah, I think that's a very good point, and I think this
It
is where
cannot be 3 hours procedure for 2 centimeter lesion.
yeah, no, exactly, and I think this is where it's important that we train the future of our endoscopists doing ESD well, because it is important to do this at a reasonable speed and efficiency, so that we're not taking a long time doing the procedure, and also as you quite rightly say, minimal complications, with these patients in terms of bleeding and perforation. Obviously, not as important in the rectum when you're below the peritoneal reflection, but
Yeah,
still.
but if you are in the beginning, sometimes it's
Yeah,
important, no?
exactly. Totally agree. Totally agree.
So tough question to you. So we have a 4 centimeter lesion with the dominant nodule and both MRI and ultrasound, they are very uncertain. So what is better? Is going through a ESD or transanal resection, full thickness with a TM or other options?
Yeah, I think it's a really good point. And what I say to our team is we need to think about what plane of dissection we're trying to achieve before we start. And that is very much reliant on what we feel the endoscopic diagnosis is and where we are in the rectum in terms of the peritoneal reflection. So what's really important is
Also anterior versus posterior as an impact?
and also if you're male or female, because if you have an anterior lesion in a female patient, even if it's mid-rectum, you may be intraperitoneal with a very, very deep pouch of Douglas. And if you're posterior, that's not going to be the case. So we need to evaluate carefully on morphology, on magnification, on position, anterior, posterior, on where we are in reflection to the peritoneal reflection. And then we can make a decision on the plane of dissection. So if we feel this is high-grade dysplasia or early submucosal invasion, the plane of dissection will be in the ESD plane, in the submucosal plane, just above the muscle. But if there's any doubt, we can be in the intermuscular plane. We try and avoid in our practice to do full thickness resection as a diagnostic tool. And let me explain why that is, and two
Yeah,
reasons.
sure, that's important.
Because one of the things that we spend a lot of time on is we want to evaluate what the patient wants to do if this is a T1 cancer with different scenarios. So we need to know before we start, and we spend a lot of time with this with the patient before, if it's a T1 cancer with vascular invasion and we've completely resected it, what do you want to do and what will you have in terms of your decision making? Will you want to have lymph node harvest? Will you want to have lymph node harvest if there's a 5% risk, if there is an 8% risk, if there's a 10% risk? Now, if they say to you, actually, I want to have lymph node harvest if this is a T1 cancer, then actually the purposes of that dissection is to get histological dissection. So we would go intermuscular for that and not go full thickness because we don't want to obscure and make it more difficult for the laparoscopic anterior section at a later stage. But if the patient says to us, actually, well, I don't want to have surgery and I want to go down the route of go down the route of local resection plus or minus potential chemoradiation after, then full thickness resection and primary closure is important with adjuvant chemoradiation. So I think that decision making is based on what the patient perceives as their risk of lymph nodes and what they want to do itself. And obviously, we spend a lot of time before we do ESD, and I do TEMS and I do transanal resections as well as ESD. So we will make that decision with the patients and help them with that process.
Okay, so you introduce a concept which may not be very familiar to all of our friends watching this talk, which is intermuscular. So can you please clarify what you mean with intermuscular? And can you tell us if we can achieve intermuscular either endoscopically or through transanal?
Yeah, I mean, I think this can be achieved, with endoscopic and with transanal. I tend to favor the endoscopic route because I just find that the views.
So can you tell us what you mean intermuscular dissection so our friends can understand? Because
So
it's a new concept for
what,
some
so
of
what,
us.
so what we would mean is we would go between the circular and the longitudinal muscle, and we would not breach the mesorectum, and therefore we would not go into the fat of the mesorectum. We're still keeping the longitudinal muscle of the rectum intact, and we're just dissecting between the planes of the circular and the longitudinal muscle.
Okay, that's a very good point. Can you tell us what is the technical trick that you need to use to maintain this plane intact and not to create any little hole or perforation?
Yeah, I think I remember initially when we first started, what we would do is we would try and develop that, get into that space early. But what we found is that if you get into that space early and you spend time, a long time in that dissection, inevitably you will split the longitudinal fibers. And we
Yeah, just with the pressure of the
just with
tip of the cap, no?
the pressure of the cap.
Yeah,
And we see that with perm as well, Ali. You see that, don't
yeah,
you?
exactly.
You'll see longitudinal. It's not as it's obviously a bit more supported in the rectum because the mesorectal fat is much more supportive than the
Hmm.
periesophageal adventitia.
On both sides, anterior and posterior, is mesorectal fat so strong?
The posterior side it's strong, and the anterior side you've got to be careful. You've got to be careful, particularly in women, because you will split, and you can then go into the peritoneal cavity if it's anterior, if it's a very deep and low pouch of Douglas.
Yeah, yeah.
So what we tend to do now in our practice is that if we've got, say, an LSTG mixed nodular and there's a dominant nodule, and we feel that that is more invasive, and we want to do a intermuscular dissection for that particular area, what we would do is we would approach this to do the ESD submucosal dissection in all of the homogeneous component. And where we feel that it's going into the nodular aspect, we would only do intermuscular dissection into that area and do that bit towards the end so that we can resect this without actually opening the longitudinal muscle and the mesorectal fat.
Can I summarize? This will be selective intermuscular.
Absolutely. Selective intermuscular rather than complete intermuscular.
That's a very good point. So thanks for clarifying this. So, I mean, this is very interesting. I will spend hours listening to you. Thank you.
I know you
So,
enjoy.
no. That's lovely. So, can we move to another topic, which is I heard you leading a very important project in the field of education for, would say, colorectal advanced endoscopic resection. It's not only the ESD concept, is much broader, like you're showing us. It's not just resecting, it's concept of indication, patients management, complications, whatever. So can you tell us what is the project, which company is behind that, what is the vision behind that?
Yeah, no, thanks, Ali, for asking that. So so I'm involved in a program with PENTAX Medical called the Forward program. The
Interesting.
concept, the concept of this is really the vision that we need to have a structured program for training endoscopists to do ESD, to make it more accessible, and to have a structured program that not only involves the educational element of the theory and the practical elements in the lab, but also to incorporate a mentorship program for endoscopists wishing to take on ESD as part of their new career. So
Okay.
it's really set up to support and mentor endoscopists who are just embarking on this journey. I think, when we started, Ali, a lot of it was about, starting and learning whilst we were doing it, and making errors as you go along, and spending a lot of time to achieve the endpoint, and spending a long time doing ESD. And what we want to try and do is shorten that training for them. Shorten, you try and avoid them making the same mistakes that we did, and to help them with tips and tricks to shorten their learning curve and to increase their efficiency of doing the ESD. So the Forward program is really based on these principles, and it's really supported by Pentax, which I'm very grateful for.
No, I'm grateful as well and delighted to hear that the company is sponsoring such innovative project. How did you select the people? It was just yourself? It was a board of people? It was Pentax?
Yeah,
How is the door to access to the project, also
so
for the future?
so the the future is such that this has now become extremely popular, and the future would be based on an application process, where we would consider, your previous experience, and also the opportunity to introduce ESD in your own institution, because it's really important that the institution supports the
Yeah,
endoscopy
extremely
to,
important. Yeah,
to
good point.
develop that procedure. Otherwise, it's not possible for them to continue with that.
You want to create a Ronaldo or Messi, but with a team around, not
Yeah,
leaving him Ronaldo playing alone in Juventus. So that's the concept, basically, no?
absolutely, absolutely. To use the football analogy, absolutely. I mean, I think, there's no point spending a long time training somebody if they're not going to have the opportunity to do the
I,
procedures in practice.
I can imagine you spend a lot of time discussing with the Pentax leadership about this project. What was your feeling with these guys?
Yeah, I mean, look, I think that what's great is that it was received with open arms in terms of trying to, have some funding to support endoscopists on this sort of long journey. And that journey is because, you know how difficult it is to attend meetings,
Very difficult. Yeah.
courses, to get hands-on experience. But even if you go and attend live courses and animal courses and workshops in the lab, even if you do all of that, when you come back to the hospital, you may forget all the skills because you don't have the opportunity, you don't have the mentorship, someone to guide you through that program. So what we actually also wanted to create was two things, hands-on and face-to-face mentorship, but also now digital remote mentorship, through
Wow,
sort of,
that's very new. So can you tell us
workshops.
about digital remote?
So what we use is we use software programs, and all our mentees will have a software where they will actually be able to record and live link with us as mentors during the procedure. And the program that we use is a program which is called Proximy, where we could actually annotate and draw on the screen while they're operating. So we can speak to them and say, look, you should be cutting here. And we can draw that on the screen so they can see what we want them to do. And then they can mimic the same movements. Now, I'm not saying that's a replacement for someone being over your shoulder. It isn't, but there are some delegates that are actually already in a bit more advanced stage and want to do the procedure in their hospital, but just need some additional support. And so that's when we would link in with them as a live link, and then mentor them through that particular.
And this is going to work? What is your feeling?
Yeah, I think it needs a lot of refinement. I think it needs a lot of refinement because it's a very, very manpower heavy program, because it involves the mentors to be available. You can imagine a lot of their time, and so I think as the program grows and we have more mentors that are available in the future, there may be an opportunity to do two things. One, to have a live mentor, and also to have some video mentorship. So what we do a lot of with some of our delegates is basically we will have them doing a case, recording a case, and then we will schedule a session where we will see their case with them, and we can discuss, what we felt they did well, what they could do differently, how could they have managed a particular bleeding area differently, and then that can give them some feedback on their actually recorded case. So I think this is a huge opportunity, and there's something that we should develop further. And I think, COVID, the time of COVID has made us develop all these different ways and novel ways of working. There's obviously no substitute for hands-on and being there in the room, but we've now looked at some novel ways of doing it, and I think it will only time will tell whether it will work. We've just
I mean,
started our first program of 12 mentees that are going through the program for the first year. And then we'll be starting our second year program in the autumn. And we can feed back to you with some data on how they've got on with their training and their implementation of ESD in their
I mean,
practice.
for the new applicants, there will be another round of mentee that you're going to select for 2022. So
Yeah, absolutely. I think we've been overwhelmed by interest. You can imagine that, lots of people have now,
you will
said
be fully booked
it was going to
quickly.
be, it needs a full-time job. So yeah, I think there's
Who
going to be
knows?
selection process, interview process, competitive entry into the program, and really dedication from the institution to
I
the
mean,
kind
I know you're using a lot of PENTAX tools for your resection procedures. I got fascinated by this new bipolar forceps for hemostasis. What is your feeling, your gut feeling?
yeah. Now, something, it's actually changed our practice, because what's interesting is that the hemostat, which is, as you quite rightly said, is a bipolar, we find it very safe in the colon, very safe
Yeah.
in the colon. And you can sort of use it with confidence without having the issue of having any deep burn to the muscle layer. So
So technically, you use it with the same approach, or you have a different technical?
it's a different approach, because as surgeons, we're quite used to using bipolar in surgery, and you need to keep the jaws slightly apart,
Mhm.
because they can work even with the jaws slightly apart.
Okay.
The other thing is that when you're using diathermy, after you've diathermied the vessel, when you open the jaws can get sticky with bipolar. So it's important to carry on using the diathermy whilst you're opening, so it goes
Okay.
away without actually having any sticky coagulum to it. So there are specific techniques that are important for you to grasp before using the hemostat. But the advantage is that it's extremely safe. It's extremely safe in the colon.
Is going to work also for spotting bleeding, or
Yeah,
we have a
absolutely.
limitation?
No, it's working for spurting bleeding as well, because obviously you can close and grasp in the same manner as you would use a monopolar forceps.
That's great.
The other advantage is, if you're not sure exactly where it is, you can keep the forceps slightly open and use,
Okay, you go wider.
yeah, and then
That's
that
a good
uses,
point.
the bipolar, and that can stun the vessel. And often we do that. What we do is we stun the vessel, slow it down, wash, and then pinpoint the exact location. So I think there's a big advantage of potentially using bipolar, and we've certainly introduced it in our practice for ESD.
Okay, I think we are running out of time, and it's been interesting, so I will spend a lot of time talking to you. So, very briefly, so where are you spending your holiday? You coming to Italy? You have any plans? Or you going to India? So, I mean, what's next for your holiday?
I wish
You, the kids, the family, where are you going, guys?
I wish I could be going to India. In fact, we've had to cancel a lot of our holidays because
Yeah, I can
the
imagine
UK
that.
is focused. But we're going to try and go to Spain in the summer if we can, in July. So I'm
Okay.
hoping to take some time off. But if I can visit Italy, if it all opens up, then that would be great.
So hopefully we will see in person very soon. We have a couple of beer
That's
to share, and thanks. Has been a great talk. A lot of innovation, a lot of new things that we discussed, and my proposal is this is not going to end. I will say probably in a couple of weeks we can do similar interview, but showing videos and showing more technical details. People will love to learn from you. And I know this is extremely important. Only outside the big program that you have with PENTAX, we can offer a huge amount of people this opportunity to receive the tips and tricks for advanced tissue resection, especially in the colorectal field. So thanks, Amin, for your time. It's been
Thank
great
you.
pleasure.
It's been a great honor. Thank you so much.
Ciao, ciao, everybody. Thank you, everybody. Thanks, Amin.